Okay, so, just before I start my presentation, I would like to say you that I'm a professor in biochemistry at the University of Pavia at the Department of Molecular Medicine, and I'm also the vice director of the tissue engineering department, and that was positively created in 2006. And and it's very good news for me to share with you the fact that also this center will become a very larger center, and the name will be not anymore a Center for Tissue Engineering, but a Center for Health Technologies. And so this will be like a strategic project from University of Pavia. Besides that, if you see, the title is quite complicated, but of course, I'm going to discuss and to present some data for different, and I will show you 4 different examples. And of course, they regard bone tissue. My presentation will be divided in 2 parts. The first part will be only on introductory slides, which are really useful to understand the experimental approaches. Bone tissue, as we say, is a major structural and supportive connective tissue of the body. We know that the function are a lot of functions: support, protection, assisting movement, storage of minerals, and the storage of energy. Besides of that, as I can show you in these slides, there are a lot of cells which are implicated in bone tissue, and as a previous speaker have already shown, some of them are the osteoprogenitor cells, osteoblast, osteocyte, osteoclast. All of them are important because they are important for the equilibrium, because bone tissue formation is always an equilibrium between cells that can create and give formation of bone tissue, and on the other side we have osteoclast with the function of resorption of bone. And then we have, of course, the other components which are important and that they are already been shown and mentioned in the morning and also in the afternoon, and of course the inorganic and organic matrix. Inorganic, especially hydroxyapatite, and regarding the organic matrix, of course, bone matrix proteins. And finally, we have the 2 membranes, periosteum and endosteum, which are 2 membranes very much important in bone tissue. Besides that, whenever we do have to approach to a tissue engineering experiments, we need to work with 3 different tools. And these tools are, of course, cells, scaffolds, and, of course, bioreactors. So, as we said, cells are really important. Of course, also the choice of the scaffold is important, but also are important the bioreactors. And so, just because in my lab, and also not only in my lab, but in our center, we are working with bioreactors, I will show you also some results that we got by using 2 different types of bioreactors. As you see from the panel, which is presented in these figures on the figure on the right, you can have 2 different ways to do a transplantation. Or let's say you can isolate cells from the patient, and then you can seed these cells on the scaffold, of course on a specific scaffolds, and then you can just use these cells plus scaffold to implant in the patient. Or eventually you can go on with the creation of a real artificial tissue, bone tissue in vitro, and then you can use this one to implant, to do tissue transplantation. Besides that, in the approach that I will show you, we have been using all of these cells which are presented in these slides. We have been using embryonic stem cells of human and murine origin. We have been using adult stem cells. Of course, in this case, we have been using especially human adult stem cells, so that means the multipotent stem cells, especially because our center is close to a hospital, so it's very easy for us to get these cells from surgeons. And finally, we have been using also induced pluripotent stem cells, either from murine or human origin. Regarding the scaffolds that we have been using, then I will show you briefly in the in the next slides the type of scaffolds we have been able to create and to characterize. But before we go on this, it is important to remind you that the scaffold properties are really important because the surface topography, the mechanical properties, or eventually the morphological properties, again electrical properties, or polymeric nanoparticles. So that means the presence in the scaffold, for example, the nanosystem or nanoparticles or nanoshells, can be important for the response of the cells to this type of scaffolds. And this is because whenever cells can interact with this biomaterial, you can have the activation of a lot of transcription factors and, of course, induction of mechanosensitive genes. And the result can be the tuning of stem cells. Finally, as we said, beside the cells, beside the scaffolds, the third important tools of a tissue engineering approach are the bioreactors. There are different types of bioreactors. I would like to show you just two examples of bioreactors that I've been using in my laboratory. One is called pulsed electromagnetic field. This is the acronym PEMF, and low-level laser intensity. Regarding the pulsed electromagnetic field, as you can see here in this first I was wondering if there is the... sorry. Thank you. So as you can see here, are indicated the parameters that I've been using for our experiment. Here you see this is the setup of the pulsed electromagnetic field. Here we do not have the controller, which is not in the figure, but of course there is a power controller. And then we have the two solenoids, which has been imposed in this way. And we have characterized all the electromagnetic field inside this space where we keep our plates with the cells and the scaffolds. And then we have been also using, as I said, another bioreactor, low-level laser intensity, and the acronym is always LLLY. And in this case, as you can see, you can have the power controller, and then you have the diode. And we have been using a wavelength of 659 nanometers to evaluate the effect of these lights on cell proliferation and differentiation. So beside the pathway that all of these biophysical stimuli performed by this type of bioreactor can have, and of course, there are differences because it depends if we have the light as for the low-level laser intensity bioreactor or the electromagnetic field in the case of pulsed electromagnetic field. What was important for us was try to understand if this type of biophysical stimuli could improve cell proliferation and differentiation. So now let's get to the, how can I say, the core of my speech. And I would like to show you 4 experimental research examples. And the first one was performed using a specific electrospoon polylactic calcium deficient hydroxyapatite nanocomposite. And we have been testing how cells, and in particular multipotent and pluripotent stem cell, could be influenced by this type of materials. Then I would like to show you a few data which are really very much interesting, because we have been able to produce a nanofiber composite in 3-dimensional hydrogel scaffolds. And of course, this type of scaffolds, we produce it to study proliferation and differentiation. But then it came out that this type of scaffold is very much interesting because it can keep the self-renewal of pluripotent stem cells. And in this case, we just focus our studies only using human pluripotent stem cells, embryonic human pluripotent stem cells, and induced pluripotent stem cells. The third example is on the biophysical stimulation of a pulsed electromagnetic field. In this case, we used nanostructured topography of the surface done by the cluster deposition of nanoparticles of titanium oxide. And we just seeded cells on these surfaces, and then we exposed the system to pulsed electromagnetic field, just to see if we could see any proliferation, any differentiation of these cells. And finally, the last example I will show you is just a few preliminary results we got on the biological effect of low-level laser intensity on human multipotent stem cell fates. I would like to remind you that the two last examples were just performed only with multipotent stem cells, so that means adult human mesenchymal stem cells, not with pluripotent stem cells. Let me start from the first example, experimental example. As we said, we have been working with some collaborators at University of Perugia, in particular Professoressa Alessandro Bianco, and she was able to prepare, as you can see, the materials. This is scanning electron microscopy images of the samples she was able to prepare, and this is a polylactic acid-based microfibrous nanocomposite, which have been loaded with different amount of calcium-deficient nanohydroxyapatite. And precisely, we have been using in the following experiments that you will see in the next slides, we have been using PLLA without hydroxyapatite, calcium-deficient nanohydroxyapatite, and then PLLA, which was loaded with 1% or 8% of calcium-deficient nanohydroxyapatite. Interesting, as you can see from this table, we could see that the storage modulus and the the loss modulus was changing from the material, and as you can see, it's very high whenever you have a PLLA, but then it becomes lower when you are using the PLLA, which has been loaded with this nanohydroxyapatite, with 8% of a nanohydroxyapatite. So on this type of materials, we have performed our experiments. So as I told you before, we have been using, so here we have the scaffolds, and the scaffolds are the ones you can see here again in the slides. And we have been using the cells. What type of cells? The cells that I mentioned you before. So that means we have been using human adult stem cells, so they mean multipotent, and we have been using also pluripotent stem cells, so that means murine embryonic stem cells and embryonic induced pluripotent stem cells. But as you can see, we performed the experiment in two different conditions, as you can see. The first one was just to use all the free type of cells and the free type of scaffolds, but in basal growth medium, so that means without any osteogenic factors. And then we have been using the condition where we added the osteogenic factors. Then we perform all the experiments, so that means we let all this system grow for almost 30 days. And then at the end of this experiment, we did again electron microscopy images, we did the quantitative RT-PCR, we did also the alkaline phosphatase assay, and also we performed the quantification of extracellular matrix proteins accumulation. And this is just the only result I would like to show you, because otherwise we can stay here forever. These data are already published 2 years ago on the Biomacromolecules journal. And as you can see here, we just show you the result we got on the quantification of proteins. And as you can see, which was very amazing for us, is that already the cells, in this case, these are the cells, the result obtained by incubating the human multipotent stem cells, already in the basal medium, you can see an increment, a real good increment of all of these proteins, which are the bone metric proteins. And this result is very interesting, if you compare to just the cells which are being grown on PLLA, and you can see that the level is even a little bit higher if you compare to the cells which have been grown on 1% calcium deficient nanohydroxyapatite, or if you compare to PLLA. But what was amazing was that the same result was obtained also using pluripotent stem cells. So we could a real good increment all of these proteins just when the cells were put it on the scaffolds without adding osteogenic factors. So what was the main conclusion of these results and these experiments was that the highest concentration of extracellular matrix was obtained and was observed on the materials with the lowest storage modulus and loss modulus. And so we came out with this graphical abstract that shows the final result of this data, showing that whenever you increment, you have this electrospoon material, which is made of PLLA. But whenever you loaded this material with calcium deficient hydroxyapatite, and also if you do an increment of the concentration of this calcium deficient hydroxyapatite, you got a very nice increment of differentiation. So that means that even if you do not add osteogenic factors, you can have a real nice increase of differentiation. So this type of scaffold, it seems to me very nice and very interesting because promote the differentiation. Let me get to the second example, experimental example we have been doing recently, and this data are just submitted to Biomaterials, and I know that they've been really the editor said that the work is very well done and the results are quite interesting. And in this case, we have been using, again, electrospoon PLLA, but the innovative technique of this material, these scaffolds that we have been able to create in collaboration with University of Milano and University of Bologna, is that we do have PLLA, which of course has been electrospinned, and then on this electrospoon material, we have dropped over these materials in hydrogel, and this hydrogel is made of oligomers. I cannot tell you much more because I'm not a chemist, and this part was, of course, performed by chemists. But I would like to show you just these scanning electron microscopy images. Here is the top view of the material, how it looks like whenever you see it, as soon as it has been formed. And then you have a transversal section that show you all the electrospoon material. But what is an innovative part of this type of scaffolds is that if you use only hydrogel is very soft. But we have been able to introduce the supporting material, which is PLLA, but using electrospoon PLLA. And also we are using, it is reported before here in this part of the legend, we have been using a nitrogen atmospheric pressure non-equilibrium plasma, which was the technique that we have used to link in a very strong way the hydrogel with the electrospoon material. So of course, when we prepare this material, we're interested to see how cells, pluripotent stem cells, could proliferate and differentiate. But we were really surprised because whenever we put these cells on this type of materials, we could see that pluripotent stem cells is either human embryonic stem cell or, as you can see here, or human-induced pluripotent stem cells. We could have on this type, the material we have been using, which is a composition, as I said, of PLLA, electrospoon PLLA, and hydrogel, are called COM10 a composite 20. They were able to grow. So if you see here just the result of proliferation at 24 hours, after 24 hours and 7 days. But whenever you see the immunofluorescence analysis just following the tubulin, beta tubulin of the cytoskeleton of these cells, we were really surprised that the cells were keeping their colonies shape, especially on the composite 10 and composite 20, both type of pluripotent stem cells. But then we want to see some more, and so for this reason we perform a quantitative RT-PCR to evaluate the classical and differentiated marker expression in human embryonic stem cells and, of course, human-induced pluripotent stem cells. Of course, this was done at 7 days, so after 7 days of seeding on this type of scaffolds. And what was amazing was that we could see a higher level of the free transfection factors, which is OCT4, NANOG, and SOX2. But as you can see, the immunofluorescence analysis was confirming the final result that if you keep these cells inside this hydrogel, you have two positive biological effects. One is that these cells can proliferate, and the second effect, which is much more important, they keep their pluripotency. And also the third and final aspect, which is really important, is that you have created the scaffolds, which is an hydrogel on one side, but on the other side, keep the support of the electrospoon PLLA. Let me get you close to the two final experimental approach. As I told you before, the first one I will show you now is just to evaluate the biological effect exerted by titanium oxide and pulse electromagnetic field on human multipotent stem cells. And here just the scanning electron microscopy of the surface at higher magnification of this titanium oxide nanostructural surfaces. And we performed, of course, the attachment analysis, and we were able to determine the Fauci area and the number of cells attached to the surfaces. And as you can see, the titanium dioxide are very nice, are useful, are very nice for the cells in the sense that cells can attach, and they have more focal point to attach to the surface. But then also when we did the morphological analysis, we could not see any differences between, this is the titanium oxide and this is glass, big differences regarding actin and beta-actin on the cells. But what was amazing is that, again, I can skip this one because it's not really enthusiastic, but we perform all the analysis that you have to perform at the end of these differentiation protocols. And I would like to show you just the table containing the quantification of the bond protein markers. And again, this experiment was always performed in basal medium, that means without osteogenic factors on the free surfaces, and with or without pulse electromagnetic field. And we could not see any specific results. But when we performed these results in the presence of osteogenic medium, we could see some differences. So we could have an increment, especially of bond protein matrix deposition for the cells that were seeded and incubated in the presence of osteogenic factors and exposed to pulsed electromagnetic field. What are the main conclusions of this story? Right now, the conclusion is that the surfaces probably is not important for the final fate of these cells. What is important is the exposure to electromagnetic field. What we can see is that probably the surfaces plus the exposure to electromagnetic field can be useful to promote cell differentiation to bone tissue. And here just a confocal analysis showing the final qualitative result that we could obtain by quantitative results. Finally, the last experiment, as I told you before, we have been working also with the low-level laser intensity. And in this case, we have performed a different type of experiments. We have been using different type of multipotent stem cells isolated but from different patients at different ages. Here, just a short summary of our results. We can see that you can use different, a single dose exposure or multiple dose exposure of laser. And we have seen that probably the exposure of 2 joule per square centimeter is the best way to get proliferation and, of course, differentiation. Of course, we did a perform apoptosis test and morphology, and we could see that cells are perfectly fine. They do not go through apoptosis, and the morphology of the cells is absolutely nice and similar to the control. And what is important is, of course, the proteins. So we also started, and these are the preliminary results, where we could allow these cells to stay for a longer time and to find out after 28 days if this multiple dose exposure can influence the production of extracellular matrix, bone extracellular matrix. And as you can see, even if these are just preliminary, we could see some increment in the bone matrix deposition. Acknowledgement, of course, we could be able to do all of these experiments thanks to all of this fund agency. I would like to thank all the people that have been involved in this type of research. Of course, here is just a photo of my small group, and beside these people, we have a lot of young students. It's a very young research group, but very eager to get nice results. And of course, thank to all of you for listening to me, and you are welcome to come to see me in Pavia.